Thank you, Christophe, for this very kind introduction, and before that, for the invitation to this wonderful day. I've been really inspired by these rapid-fire sessions this morning. I can also see why you placed me now in the program between this wonderful reflection we just had on translational research and the soon-to-come cancer session. I hope I can bridge the two. Bridging is my business. I'm a translational researcher, so the story I'd like to tell you about today is the rise of engineered T lymphocytes as cancer drugs. I think we all know that a key feature to reconcile the objectives of safety and efficacy in the development of cancer drugs is their specificity. And it turns out nature offers us exquisite ways to achieve specificity through the adaptive T-cell immune system. T-cells are not only highly specific, but they can be persisting or long-acting, if you want to describe them as a drug. However, they often lack the potency that they would need to eliminate the tumors, recognize the tumors, and overcome a number of obstacles that T-cells encounter in the tumor microenvironment. And so, as you know, the T-cells are born in the thymus, from where they get their T, and acquire, through the process of VDJ gene recombination, a unique receptor for antigen that is characteristic of every clonotype. Our repertoire, the sum total of all our T-cell specificities, is, however, large but not infinite. And so the idea that you could instruct T-cells to recognize any antigen of your choosing and potentially overcome tolerance to self is the underpinning, really, of this idea that you could engineer T-cells for therapeutic purposes. And the paradigm, perhaps, for this is what we call CD19 CAR Therapy, which I'll describe in the next few slides. CD19 CAR Therapy is born at the intersection of different fields. In the upper right there, I recall that it relies on fundamental discoveries in basic T-cell immunobiology that teaches us about antigen recognition and clonal T-cell expansion. And today it continues to teach us much about the composition of the tumor microenvironment. But in the upper left here, I also want to point out that this emerging discipline, is heavily predicated on genetic engineering, which started in the beginning with gamma retroviral vectors and today extends to gene editing. In the middle right, here I introduced the notion of synthetic receptors, which we termed CARs, or chimeric antigen receptors, some years ago to provide such a genetic instruction to T cells. And the middle left here points out to the need to identify particular kinds of targets for these receptors, which, unlike the physiological receptor for antigen, the T-cell receptor, are not HLA peptide complexes, but any molecule that you can find on the surface of a cancer cell, may it be a protein, a glycolipid, or a carbohydrate. The lower right is to say that to convince the world that this academic exercise had any value, it was necessary to bring it to the clinic and to show that it can be effective. And therefore, the few initial players in this field had to also develop cell manufacturing sciences that could eventually advance to the clinic, and that's the lower left, by meeting the strict regulatory requirements imposed in the US by the Food and Drug Administration. So how to go through 30 years of history in one slide? Well, I'll just say that as was just said, I went to MIT some years ago to figure out first how to introduce a gene in T cells. It took 3 years to get that little result shown up there, which shows you in one panel a few percent of T cells expressing the E. coli LAC-Z gene. Today, you can teach a high school student in one afternoon to introduce as many genes as they want, really, in a cohort of T cells. We then had to focus on the design of receptors to instruct the T cell to recognize the antigens of our choosing. There had been many initial efforts, in particular one called a T-body, a fusion between the activating chain of the T-cell receptor complex called the zeta chain and scFvs derived from immunoglobulins, which could serve to direct the cytotoxicity of T cells. But because we had the means to evaluate such molecules not only in clones or leukemia cells, but primary T cells, we soon realized that this design was not sufficient to produce a productive immune response. And for that, we had to incorporate, shown into the far right there, an additional signal, a costimulatory signal, thus creating dual signaling molecules that we termed at that point second-generation CARs. In the lower left here goes to our selection of targets, initially for mouse studies, but one that we thought we could in due course bring to the clinic. And in the late 90s, we set our eyes on a molecule called CD19. It is a cell surface molecule, and it is found on the vast majority of leukemias and lymphomas, for which there was blatantly a major unmet medical need. And this diagram here is just to show that some 16 years ago now, we reported that you could take human peripheral blood T cells, instruct them through genetic engineering using a gamma retroviral vector that encoded a chimeric antigen receptor specific for CD19. And following a single infusion of human peripheral blood T cells engineered in this manner, you could treat and eventually cure a range of B-cell malignancies in mice bearing these tumors. There was no industry to turn to in those years, and so academic centers had to take it on their own to establish the methods and the principles for human T-cell engineering. And so my colleague Isabelle Rivière at Memorial Sloan Kettering set out to first construct the GMP facility and then develop this manufacturing process that you see here, in which T cells are collected from the peripheral blood of cancer patients through an apheresis and then genetically modified using gamma retroviral vectors. You can use other devices such as lentiviral vectors or DNA transposons today, culture the cells for about 10 days in the bag that's shown in the lower right, and then after some QC, quality control release testing, freeze down these cells or infuse them immediately if needed. So we undertook these clinical trials starting around 2007, 8, 9, and two other groups at the same time as us who got interested in our work also undertook clinical trials at the National Cancer Institute and at the University of Pennsylvania. But on the left here, I'll show our example. That was the first report of the efficacy of this treatment in adult patients with refractory and relapsed acute lymphoblastic leukemia. In those FACS profiles on the lower left that you see here, you can see this disease of CD19 positive relapsed ALL in two subjects here, 4 and 5. You see the blasts up above here. You can see their prompt disappearance following the infusion of T cells. These qualify as complete responses, and even more than that, the tumors soon became undetectable by deep sequencing, thus qualifying these responses as molecular complete responses, something that we actually, I have to say, had not even expected for a phase 1 result. The NCI was the first to report a patient with non-Hodgkin lymphoma treated with the CD19 CAR, and the University of Pennsylvania was the first to report on 3 patients with chronic lymphocytic leukemia treated with this. And in this study, we reported on 5 patients with relapsed ALL. Later that year, in 2013, we were, of course, elated that the Journal of Science would proclaim cancer immunotherapy to be the scientific breakthrough of the year. Now, the lion's share of that was for checkpoint blockade, a more mature form of cancer immunotherapy. But we were, of course, thrilled that the editors also recognized this emerging immunotherapy based on a cell-based medicine. Now, since then, I think you all know that these results, these very dramatic results obtained in the very first phase 1 clinical trials were reproduced in a number of larger trials. So at some point in an interview with the New York Times some years ago, as the journalist was asking me, well, what do you exactly do? You're talking about viruses, synthetic receptors, expansions of cells that sometimes grow inside the body of the patient. What do you do? And this is when the term was, well, what do we do? Well, we create living drugs. And this living drug has to recognize the antigen, kill its target, but also multiply upon encountering its target. And multiplication, proliferation is not enough. It has to remain functional and sustain its function over time. So how to show you a snapshot of first, the FDA then approved these first CAR therapies in 2017. Both of them are specific for the antigen CD19. Both of them are second-generation CARs. They, however, differ in the usage of the costimulatory domain that is incorporated in these CARs. To the left is one that uses the CD28 domain. This is the one originally described by us at Sloan Kettering, later used at the National Cancer Institute, and today commercialized by a company called Kite Gilead. To the right is a second-generation CAR using the costimulatory domain from another costimulatory receptor named 4-1BB, a member of the TNF receptor family. And it too has been used in first young patients with ALL and is commercialized today by Novartis. So a snapshot of the state of the art, if you like, with these molecules in just one slide summarizing from four clinical studies. I'll start with our own. Since last year, we updated on the first 53 patients that we had treated with CD19 CAR T cells, all adults with relapsed refractory disease. And you can see the good news that some of these patients will enjoy long-term benefits in the form of survival, and I think we can now begin to proclaim some of them to be cured. Likewise, from the Children's Hospital of Philadelphia, you can see in children with ALL, it's a different time scale, here only up to 22 months. And again, very high complete remission rates, like in adults, on the order of 85 to 90%. But I have to say that here too, about half of the patients will eventually relapse. This study uses a CD28-based CAR in adults. This study uses a 4-1BB-based CAR in children. And below are two large studies in non-Hodgkin lymphomas, another form of CD19-positive cancer. And to the left here, a multi-center study making use of a CD28-based CAR. And the complete remission rate here is a bit less than ALL, but then the relapses still present also are a bit less. And here to the right, a study from the University of Pennsylvania using a 4-1BB-based CAR in patients with non-Hodgkin lymphoma. And here too, again, about a 60% complete remission rate with a third to a half of the patients that will eventually relapse. So good news, because all of these patients had refractory B-cell malignancies and no treatment option to look forward to. At the same time, you see the need to still improve these therapies. And this is important not only for the B-cell malignancies, but in order to bring this technology to treat solid cancers. So what can we do about that? And I'm going to discuss a few examples of our more recent work, aiming to generate yet a better chimeric antigen receptor T cell. So let me first introduce the model that we use for this. It's a mouse model that we call a CAR stress test. It's an NSG mouse, a highly immunodeficient mouse, in which we introduce a CD19-positive ALL, typically the NALM6 cell line. And then we treat these mice with CAR T cells at low doses. The point of the low dose is to stress the T cell, make it difficult for the T cell to find the tumor and eventually amplify in numbers and catch up to the tumor and eliminate it. So these, again, are the two CARs used in this study here, which are those that are FDA approved. In red here, the CD28-based CAR, and here the 4-1BB-based CAR. At these doses of 400,000 CAR T cells per mouse or less, you can see that there is no long-term cure achieved in these mice. You could do so at a higher dose, not shown here. It's very informative to go and study these mice and see what happens to the tumor in the CAR T cells. And here's what you would see examining these mice at days 7, 14, and 21 after the infusion of T cells. So first, the enumeration of CAR T cells in the bone marrow, which is the site of disease. You can see the two second-generation CARs have accumulated to about comparable numbers, far more than the first-generation CAR that lacks any co-stimulation. Their impact on the tumor, however, is very different. The CD28-based CAR has essentially already eliminated the tumor by day 7, but not the 4-1BB CAR, present in similar numbers it has not yet eliminated the cancer. And these data corroborate many others that we know, that the CD28 CAR is a very powerful effector, and the 4-1BB CAR is a milder effector. Now, over time, you can see that this CD28-based CAR is rapidly diminishing in cell numbers and eventually waning, in contrast to the 4-1BB-based CAR. If you look closely, you will note that the cell number is higher here on day 14 than it was on day 7, and it too that will eventually wane over time. And so, while the 4-1BB CAR is less potent, it persists longer, and it too eventually eliminates the cancer in these mice. So I walked you through this in some detail because these data recapitulate observations in numerous clinical trials. Of course, it's difficult to compare trials that use slightly different CARs, different manufacturing methods, different patients, different diseases. But here you see side by side in the same donor, in the same experiment, these differences in potency and persistence between these two CARs that are still both effective. So what would you do if you wanted to improve these CARs? Well, if you wanted to focus on the 4-1BB CAR, you might say, well, how could we make this persisting CAR T-cell more potent? Or if you looked at the CD28 CAR, you might say, well, how could we make it persist longer? And we chose to focus on the latter. And now I should remind you that when the normal physiological T-cell receptor, which is shown here, engages antigen, it signals through this so-called CD3 complex that comprises this zeta chain that I already mentioned. Soon as the synapse forms between the T-cell and its targets, this CD28 costimulatory molecule is called into the synapse, typically at a ratio of about 3:1. Obviously, if you look at this synthetic receptor, the CAR, you'll see that the stoichiometry is different, 1:1, because this is a covalent bond. And of course, the timing of the activation and co-stimulation cannot be quite identical to the physiological one. Moreover, extensive literature in the mouse tells us that the signals coming from CD28 and CD3 zeta are in part overlapping. And so we started thinking that perhaps we had created a molecule that was overpowering the T-cell. And we thought we might ask this question by calibrating the strength of activation and introducing mutations in this CD3 zeta chain domain. We know from now classic literature that it comprises these 3 regions called ITAMs, these activation motifs, and so introduced mutations creating, for example, 1XX, in which the first ITAM is still functional and the next 2 have been inactivated, or others, and for example here XX3, which retains the third ITAM but lacks the first 2. So we go to the stress test again, and what do we find? Same dose, rigorously measured and infused in mice on the same day, of course. Well, again, at this dose, the 28Z CAR, the 3 ITAM classic CAR, is borderline in efficacy. There's tumor progression over time. The XX3, which has a single ITAM in position 3, induces responses, but then all mice relapse. In contrast, this other 1 ITAM containing CAR induces responses that are durable. Note the tumor progression that we want to see every time, because that's the essence of the CAR stress test. It's a low T-cell dose. The T-cell has to migrate to the marrow, find the tumor, get activated, begin killing, expand, maintain its function, and eventually catch up to the leukemia and eliminate it. So what do we think this calibration of signal can do? Well, I'll just show you one experiment here that tells us that the CARs not only determine the potency and persistence of T cells, but can also determine their fate, meaning their differentiation. So in this experiment, we start from human peripheral blood T cells, this time purify those so-called naive T cells that have never encountered antigen before. We then introduce the CAR into what's called the track locus using CRISPR-Cas9, which I'll introduce in the next slides, such that all of these CARs are placed under the same transcriptional control inside these human naive T cells. We then expose them to antigen CD19 such that these naive T cells now signal for the first time through the CAR. And a few days later, we extract RNA and query what is their transcriptional profile. And we compare it to controls here that are shown on the left, where we reproduced work from Luca Gattinoni at the NCI, who characterized these profiles in different T-cell subsets. On the left here is an effector profile, or the profile of effector T-cells. All these genes in red here are effector genes, and all the genes that are off here are memory-related genes. In contrast, a naive T-cell preserves its memory circuit, while its effector genes are turned off. So what do we find in our CAR-engineered peripheral blood T-cells? Well, the 1928Z CAR is shown here. And you can see beautiful effector function is no more memory. It fits this clinical picture that I showed you of a powerful T-cell that has terminated its renewal programs. The XX3 that I showed you before, which failed to control the tumor, indeed has its memory circuits on, but the effector functions are minimally activated. And it is a poor therapeutic CAR T-cell. And the 1XX has this hybrid profile that's shown here. And you recall that it controlled the best the T-cell. And so we interpret this to mean that with calibration of strength of activation, as shown here in 1XX, the strength of signaling is sufficient to turn on therapeutic effector functions, but not so strong as to turn off the memory function, therefore allowing for the greater persistence of this CAR T-cell. And obviously, this 1XX CAR is now the one that we are going to bring in multiple clinical trials at Sloan Kettering starting next May. With the advent of these amazing tools for targeted gene integration, we could now ask the question, what difference does it make where the CAR gene, the CAR cDNA, integrates in human peripheral blood T cells? And I'll show you here data obtained with one particular targeted integration, where this 1928Z CAR is now introduced into what's called the TRAC locus, the locus that encodes the T-cell receptor alpha chain. And this is the AAV vector that's used for homologous recombination to this site. And you'll note that it lacks a promoter. We are therefore placing the transcriptional regulation on this CAR into the human genome using the V-alpha promoter. Again, a stress test comparing the classic CAR T-cell engineered with a retro or a lentiviral vector to the TRAC-engineered T-cell. You can see the difference here at these different doses of 200,000 or less. At this dose, this CAR T-cell, which works in patients, obviously, and works at higher doses, here is ineffective. And that's in contrast to the TRAC CAR T-cell that you see here, which induces these durable responses with these gains in survival. So clearly, it's a more powerful T-cell. An interesting control, I believe, is this one here, where we disrupt the T-cell receptor alpha locus using the same guide RNA and CRISPR-Cas9, but introduce the CAR using a randomly integrating vector, a retroviral vector. And you see there's no benefit here. And so we conclude from this that the improved function of the TRAC gene-edited CAR T-cell comes not from the deletion of the endogenous T-cell receptor, but from having placed it under the transcriptional control of that endogenous promoter. And so why is this? Well, what we find in these mouse studies is that this figure here, this work is published, I'm just showing you some excerpt here. By day 17, many of the conventionally engineered T cells are starting to silence. You can see that in their transcription factor profiles, or in the fact that a large percentage of them in black here co-express these three markers, PD-1, LAG3, and TIM3, which are signatures of T cell exhaustion. But the edited T cell has not begun to exhaust. And why might that be the case? Well, the nature of T-cell exhaustion is still the object of considerable basic research, but one observation we made is shown here, which we think contributes to this. So we take our CAR T-cells and expose them to antigen in vitro, and so to antigen at time 0. These are all TRAC-edited CAR T-cells. And we observe the CAR on the surface of the T-cell. So here's the TRAC CAR T-cell at time 0. 12 hours later, you can see that the CAR has virtually disappeared from the cell surface, and then it reappears to exactly the same level as before. And that's in contrast to the retro or lentivirally engineered T-cell. Broader peak here because of variation in vector copy number and variegated gene expression, poor internalization, and then overshooting expression because those promoters are responsive to T-cell activation. Averaging a number of experiments, you see this curve here. In the track-edited T cell in blue, you can see the diminution and the CAR coming back to the surface at a certain pace. The lentiviral engineered T cells, or retroviral, you can see don't completely internalize. This is weakening, but in red, and you can see then this overshoot of the CAR. So the CAR promptly comes back to the cell surface, which means that the cell is ready to kill again. And in fact, it can serially kill many times in the short term. And our interpretation of these data, though, is that the track-edited CAR T cell that kills and then takes a break and then kills again, takes another break and then kills again, is less prone to T cell exhaustion, in contrast to a cell that kills very rapidly. And we all know that for a T cell to eliminate the cancer in a patient, it will have to remain functional for at least several weeks. And so we are getting close to bringing this to the clinic. We have now at Sloan Kettering in our GMP facility established the manufacturing process. We are still waiting for the clinical grade AAV vector in order to open the trial, and we hope to test this hypothesis that track-edited T cells are less prone to exhaustion, and that therefore they can achieve the same results using at a lower dose, and that if we use a lower dose, there will be also fewer of the toxicities that are associated with massive CAR T cell expansion. We're trying to learn from the failures, of course, of CAR therapy, and a number of mechanisms have now been identified that contribute to relapses in patients. Exhaustion, I just discussed, the premature, if you like, diminution of the function of CAR T cells. Some relapses are antigen negative, CD19 negative, but others still have some CD19 on their cell surface. And sometimes the T cells are still present. And so we wanted to know, to better understand, why might this kind of relapse occur? So again, these stress tests, you can see the effects here of infusing, lowering T cell doses, and at this dose, again, we see these relapses more so with the BB than the 28-base CAR. And now here we will focus on these relapses. So not shown because of time is the fact that in these relapsing 4-1BB CAR T cell treated mice, there still are plenty of T cells. But here's what we observe when we look at the leukemia itself. And here we are quantifying the level of antigen, CD19, on these leukemias. These leukemias in mice have about 10,000 CD19 molecules per cell. And this is what we see in the 4-1BB relapsing mice, these mice that are up here. Lower levels of CD19, 5,000, 4,000, sometimes 1,000 or 800 molecules per cell. And the T cells, not shown on this slide, are still there, as I mentioned before. But they're failing to kill because the tumor is progressing. So we wondered, did the CAR T cells select for negative variants? Would we find mutations, for example, that would account for this relapse? Well, as we were embarking to do that, we observed that when we remove these CAR T cells from this relapsing mouse and put them back in culture, soon CD19 reappears on the cell surface. It's higher, about 20,000 molecules per cell in culture, higher than in vivo. And you can see that over about a week, these very same tumors here on day 0 coming out of the relapsing mouse recover their CD19 expression. Also, we had noted that these tumors with diminished CD19 expression had normal levels of RNA, suggesting that the mechanism is post-transcriptional. And so we established a co-culture system where we co-incubate the CAR T cell and the leukemia and started quantifying the expression of the target antigen over time. And as you see here, within hours, CD19 expression decreases on NALM6, but at the same time, you see it appearing on the T cell. And because this was repeated with a CD19 M-cherry gene fusion, we also see the T cells becoming M-cherry positive on the far right. That means that the complete CD19 M-cherry fusion molecule is extracted from the tumor and incorporated into the T cell. To really confirm that this was trogocytosis, the acquisition of a piece of membrane by the T cell, we performed this experiment in which the tumors were cultured for a week with heavy amino acids and the T cells with normal amino acids, repeat this co-incubation, separate the M-cherry positive CAR T cells, perform quantitative mass spectrometry, and confirm, of course, that CD19 has been transferred to these cells, as well as hundreds of other molecules. The T cell is literally feeding on the tumor. It is taking a big chunk of T cell and incorporating it in itself. And other molecules, and CD81 is one that we highlight here, which is associated with CD19 on the tumor cell, also is now one of those molecules appearing in the T cell. So there are many consequences of this, fratricide killing and others that I don't have to walk through, but I'll just show you quickly how this informs our therapy, because that is still our purpose. So one thing you might say, well, these mice are relapsing, let's give them a second dose of CAR T cells. Well, when we do that, we treat them with the 4-1BB, which we know is almost a guaranteed relapse at this T cell dose, retreat them on day 10. A second infusion of these 4-1BB CAR T cells, shown in red and orange there, is of no benefit. The 28Z CAR, which detects a lower level of antigen than the 4-1BB CAR, does a better job, although it too will eventually fail. And that's because we know that there's a limit to the sensitivity of antigen detection of CAR T cells. This is illustrated here, where we create, through genetic engineering, CD19 target cells with different levels of antigen. If you have 10,000, 5,000, or 1,000 molecules per cell down to low, the CAR T cells work. But if it's lower, the T cells cannot recognize. So I will go very fast here because I talk too much and I know we have time for questions, so I will just skip this notion that dual targeting, having two CARs that recognize two separate antigens two CARs in the same T cell, can improve these outcomes, although there still is a limit of detection, and I'm afraid I will not go into this. I want to present a quick story in 2 minutes. And it's a new direction, our future direction of our work. The success of CD19 CAR T cell therapy, I think, has inspired many to explore other cell types as the basis for therapies of this kind. And another question is whether we could not rely only on autologous T cells, as the field has done to this date, but generate CAR T cells from alternative donors. And there's a number of cells that are being explored today, either cells from healthy donors that can be genetically modified in one way or another, or cells that can be generated in vitro from cord blood, or something that we are doing, which is trying to generate the CAR T cell in vitro, starting from the iPS cell. So this is just a concept that I want to show you. The question is this: can we produce induced pluripotent stem cells, reprogram to a ground state of pluripotency, introduce CARs, other modifications on their HLA and their functional properties, and rederive functional T cells? Well, there's a certain number of basic questions that have to be addressed here to be able to recapitulate this differentiation in vitro. And we provided some evidence in support of this approach in a publication a few years ago, and continue to work on this. As I said, I don't have time to show you much data, but I will show you that it is today possible to derive from an iPS cell a T cell that lacks a T cell receptor, but only has the CAR as its sole determinant of activity on its cell surface. These cells kill in vitro, and at least on a good day, we can generate cells that have the hallmarks of naive T cells. And why would we want to do this? Well, these would be CAR T cells born in a dish, produced in a bioreactor, and not collected from a human being. And the potential advantage of this is, of course, to control their antigen specificity, to have clean genetic modifications, and of course control, as needed, their histocompatibility. So CAR T cells, in my opinion, represents a third way in medicine to direct T cell responses. The most classic is achieved through active immunization. The more recent one in oncology is one where you de-repress latent immune T cell responses through the use of checkpoint blockade. And the third one is the one that I think CAR T cell therapy illustrates, which is a synthetic T-cell response obtained through genetic engineering, which I would call induce, de-repress, and instruct. And where should we apply this? Well, we think that one particular area of interest are those cancers that have low mutation burden and where checkpoint blockade is unlikely to work. And furthermore, we believe that CAR T cells will find many more applications beyond cancer, not only in CD19 diseases, other hematological malignancies, and hopefully solid tumors, but autoimmunity and severe infections. And Christophe, I'll stop here. Sorry.
Thank you very much for a fantastic presentation. I think you can see if you are a genius and then you specialize in something really, really well for a long time, you can get very, very far. Let me mention that you were also, I think, among the co-founders of Juno, the company that took this commercial and was eventually sold to Celgene for, I think, was it 10 billion?
Yes, for them, not for me.
Yeah, I'm afraid so. So my first question would have been, do we ever have to worry about grants anymore? You still do, right?
Oh, yes, yes.
Yeah.
But let's not go there.
Yes.
We did co-found with colleagues at the Fred Hutchinson Cancer Center in Seattle, this company, Juno. But as is often the case, they do things on their own. They may not necessarily listen to their co-founders.
Yeah, it's true. But I think that is still something that needs to be fixed in the system, that scientists like yourself do not have to worry about funding after something like this anymore.
Oh, that's nice to hear.
Yeah. Questions?
Thank you for the great talk. I have two quick questions. So first one, you mentioned toxicity, and in light of emerging reports for side effects, is there a version of CAR T cell with an on and off switch on the horizon? And the second question, how far are we to see beautiful results for solid tumors with CAR T cells?
Great questions. Well, so I had to pick my topics, and I did not address toxicity. One of the toxicities that occurs soon after the CAR T-cell infusion is what we term cytokine release syndrome, which is characterized by high fevers persisting and often leading to hypotension, sometimes requiring protection of airways in the patients and transfer to the intensive care unit. Initially, there were a few tragic outcomes in the very first trials. When this occurred and surprised us all, as this had not been seen in mouse models, but clearly it was seen in patients. Today, there are means to control this cytokine release syndrome, which, by the way, is the hallmark of a T-cell response. You know, if you have an infection, you know, it's not the virus, but your T-cell response that's inducing the fever and other effects. But in some of these patients, this response is very intense. So it can be mitigated with the use of a monoclonal antibody blocking the IL-6 receptor and corticosteroids. And it's for this reason that these approved drugs can only be administered in hospitals where the personnel, the staff, has been trained to handle this complication, which today is handled at very high success rate. But ultimately, what we want is not to have a means to only react to the CRS, but to prevent it. And to prevent it, we need to understand it. And to understand it, we need models. And it took us a while to finally get a mouse model of this. And what we found in the model is that this cytokine interleukin-6, which is not the only contributor to the CRS, but a critical one, is not made by the CAR T cell. Yes, CAR T cells make some IL-6, but most of it comes from other cells in the recipient. This was in the mouse, and these are macrophage. So the CAR T cell migrates to the tumor, and upon its activation, also in collateral fashion activates macrophage, which are the source of IL-6, IL-1, and other things that can be detrimental. So as we understand better now, this opens new venues for trying to control this CRS. One that we described is the use of IL-1 receptor antagonists, or anakinra. Anecdotally, I hear now that it has been used in a few patients, and if it is used early because it's prevention or early intervention, it can be effective, and it crosses the the blood-brain barrier, which the IL-6 receptor antibody does not do. So I think there will be other improvements to this, and this will be pivotal to allow the broader use of this therapy and outside of the hospital. Solid tumors, well, it's a big topic. Historically, these CARs have been developed with a focus on leukemia and lymphoma. I think they open the path, but that they are not sufficient. Other than finding the right targets, we know that T cells will face a more complex and more challenging tumor microenvironment, and this applies to any immunotherapy. I think that the uniqueness of a T cell-based approach, of a cell-based approach, is that you can engineer your T cell to do much more. So we are now multiplexing changes and creating CAR T cells that secrete molecules, of course cytokines, but also scFvs. One of my former students now at MGH has recently published on a CAR T cell that secretes a dual engager, so as to amplify the tumor immune response. And I think that through the augmentation of these properties, hopefully we will be able to tackle solid tumors.
I think we can do one more. Okay, over there.
Dr. Sadelain, excellent talk, very inspiring. You said your job was to build bridges, and you have built bridges both in Paris and in the US. So from your point of view, what's most important from this systems perspective, enabling scientists, translational scientists like you to build great bridges fastly to get drugs into the hands of patients?
I might have missed the exact question then. What is the...
So, translational medicine depends on systems, and we have different systems in Europe and in the US.
I see. Okay, I'm sorry. Yeah. I think I had missed that. I'm sorry. I think this therapy is relatively radical. I'll use a word, subversive. I don't think it's the way that certainly the pharmaceutical industry had been thinking until recently to intervene in cancer. You know, traditionally the pharmaceutical industry is great at producing chemicals, drugs, and then took on board proteins, you know, antibodies, recombinant factors, sometimes proteins as vaccine extracts, but never went into cells as medicine. But it's not just the pharmaceutical industry. I think the medical establishment tends to be conservative and still prefer to go with what works. And I have to say, it has not been easy to fund this work. In fact, the early part was supported by NIH, but as we got closer to the clinic, the NIH funding melted. I mean, we didn't get our grants. And so I have to say that what allowed us to advance was largely philanthropy and the support of foundations. And I don't know if this would extend to Europe, but in the US and in New York City, which is in part the reason why I went to that city, I knew that we could access funding from supporters who were more willing to take risks. And that's how we got the first trial off the ground. And so I think this was a critical difference. My center also is a big experimental center. The Memorial Sloan Kettering Cancer Center is the third holder of INDs, investigational new drug applications in the US. The first is the NIH. The second is University of California, which is everybody in California. Stanford, UCLA, UCSD, et cetera. And the third is Sloan Kettering. Now, don't think that they all loved us. We had trouble putting patients on the first trials, but I think that we have to acknowledge philanthropy. The other thing that I didn't hear, that I would just add to the session that preceded on translational research, how to best support it, is that this is team science. And the team science requires that you aggregate complementary expertise that no single human being can have. And that requires the ability to recruit staff with certain expertise, and as we heard before, who are not recruited and promoted on conventional criteria, which often have to do with their independence. Can you function on your own? If you're independent, you deserve to be promoted. Well, in team science, there's a lot of interdependent research, and there are extremely valuable people who may not be independent, but still are crucial to that success. And although it wasn't easy, I have to say that my leadership eventually agreed to hire people of this kind, who are to me as good as the best of the independent scientists, and that was also critical in moving this forward.